Evolution of sexual reproduction. Unsolved problem in biology. What selection? Pressures led to the evolution in. Evolution of sexual reproduction describes how sexually reproducing animals, plants, fungi and protists could have evolved from a common ancestor that was a single-celled eukaryotic species. 1. 3. Reproduction is widespread in eukaryotes, though a few eukaryotic species have secondarily lost the ability to reproduce sexually, such as Beteloidea, and some plants and animals routinely reproduce asexually, by apomixis and parthenogenesis without entirely having lost sex. The evolution of sexual reproduction contains two related yet distinct themes, its origin and its maintenance. Bacteria and prokaryotes have processes that can transfer DNA from one cell to another: conjugation, transformation, and transduction. 4. But it is unclear if these processes are evolutionarily related to sexual reproduction in eukaryotes. 5. True sexual reproduction by meiosis and cell fusion is thought to have arisen in the last eukaryotic common ancestor, possibly via several processes of varying success, and then to have persisted. 6. Ladybirds mating. Pollen production is an essential step in sexual reproduction of seed plants. Hypotheses for the origin of sex are difficult to verify experimentally. Outside of evolutionary computation, most current work has focused on the persistence of sexual reproduction over evolutionary time. The maintenance of sexual reproduction, specifically, of its dioecious form, by natural selection in a highly competitive world has long been one of the maintenance of sexual reproduction. More unsolved problems in biology. Mysteries of biology, since both other known mechanisms of reproduction, asexual reproduction and hermaphroditism, possess apparent advantages over it. Sexual reproduction can proceed by budding, fission, or spore formation and does not involve the union of gametes, which accordingly results in a much faster rate of reproduction compared to sexual reproduction, where 50% of offspring are males and unable to produce offspring themselves. In hermaphroditic reproduction, each of the two parent organisms required for the formation of a zygote can provide either the male or the female gamete, which leads to advantages in both size and genetic variance of a population. Sexual reproduction therefore must offer significant fitness advantages because, despite the twofold cost of sex, see below, it dominates among multicellular forms of life, implying that the fitness of offspring produced by sexual processes outweighs the costs. Sexual reproduction derives from recombination, where parent genotypes are reorganized and shared with the offspring. It stands in contrast to single-parent asexual replication, where the offspring is always identical to the parents barring mutation. Recombination supplies two fault-tolerance mechanisms at the molecular level: recombinational DNA repair, promoted during meiosis because homologous chromosomes pair at that time, and complementation, also known as heterosis, hybrid vigor or masking of mutations. Reproduction, including modes of sexual reproduction, features in the writings of Aristotle. Modern philosophical scientific thinking on the problem dates from at least Erasmus Darwin, 1731–1802, in the 18th century. 7. Darwin picked up the thread in 1885, arguing that sex serves to generate genetic variation, as detailed in the majority of the explanations below. 8. Charles Darwin, 1809–1882, concluded that the effect of hybrid vigor complementation is amply sufficient to account for the genesis of the two sexes. 9. This is consistent with the repair and complementation hypothesis, described below. Since the emergence of the modern evolution synthesis in the 20th century, numerous biologists including W. D. Hamilton, Alexei Kondrashov, George C. Williams, Harris Bernstein, Carol Bernstein, Michael M. Cox, Frederick A. Hoff and Richard E. Mykid, have suggested competing explanations for how a vast array of different living species maintain sexual reproduction. Concept of sex includes two fundamental phenomena: the sexual process, fusion of genetic information of two individuals, and sexual differentiation, separation of this information into two. Historical perspective. Advantages of sex and sexual reproduction. Parts. Depending on the presence or absence of these phenomena, all of the existing forms of reproduction can be classified as asexual, hermaphrodite or dioecious. The sexual process and sexual differentiation are different phenomena, and, in essence, are diametrically opposed. The first increases diversity of genotypes, and the second decreases it by half. Reproductive advantages of the asexual forms are in quantity of the progeny, and the advantages of the hermaphrodite forms are in maximal diversity. Transition from the hermaphrodite to dioecious state leads to a loss of at least half of the diversity. So, the primary challenge is to explain the advantages given by sexual differentiation, i.e. the benefits of two separate sexes compared to hermaphrodites, rather than to explain benefits of sexual forms, hermaphrodite plus dioecious, over asexual ones. So, the primary challenge is to explain the advantages given by sexual differentiation, i.e. the benefits of two separate sexes compared to hermaphrodites, rather than to explain benefits of sexual forms, hermaphrodite plus dioecious, over asexual ones. It is understood that since sexual reproduction is not associated with any clear reproductive advantages over asexual reproduction, there should be some important advantages in evolution. 10. Advantages due to genetic variation, DNA repair and genetic complementation. For the advantage due to genetic variation, there are three possible reasons this might happen. First, sexual reproduction can combine the effects of two beneficial mutations in the same individual, i.e. sex aids in the spread of advantageous traits, without the mutations having to have occurred one after another in a single line of descendants. 11. Sex acts to bring together currently deleterious mutations to create severely unfit individuals that are then eliminated from the population, i.e. sex aids in the removal of deleterious genes. However, in organisms containing only one set of chromosomes, deleterious mutations would be eliminated immediately, and therefore removal of harmful mutations is an unlikely benefit for sexual reproduction. Lastly, sex creates new gene combinations that may be more fit than previously existing ones, or may simply lead to reduced competition among relatives. For the advanced DNA repair, there is an immediate large benefit of removing DNA damage by recombinational DNA repair during meiosis, assuming the initial mutation rate is higher than optimal 12, since this removal allows greater survival of progeny with undamaged DNA. Advantage complementation to each sexual partner is avoidance of the bad effects of their deleterious recessive genes in progeny by the masking effect of normal dominant genes contributed by the other partner. 13-14. The classes of hypotheses based on the creation of variation are further broken down below. A number of these hypotheses may be true in any given species, they are not mutually exclusive, and different hypotheses may apply in different species. However, a research framework based on creation of variation has yet to be found that allows one to determine whether the reason for sex is universal for all sexual species, and, if not, which mechanisms are acting in each species. On the other hand, the maintenance of sex based on DNA repair and complementation applies widely to all sexual species. Protection from major genetic mutation. In contrast to that sex promotes genetic variation, Heng, 15, and Gorelick and Heng, 16, reviewed evidence that sex actually acts as a constraint on genetic variation. They consider that sex the coarse filter, weeding out major genetic changes, such as chromosomal rearrangements, but permitting minor variation, such as changes at the nucleotide or gene level, that are often neutral, to pass through the sexual sieve. Novel genotypes. This diagram illustrates how sex might create novel genotypes more rapidly. Two advantageous alleles A and B occur at random. Two alleles are recombined rapidly in a sexual population top, but in an asexual population bottom, the two alleles must independently arise because of clonal interference. Sex could be a method by which novel genotypes are created. Because sex combines genes from two individuals, sexually reproducing populations can more easily combine advantageous genes than can asexual populations. In a sexual population, two different advantageous alleles arise at different loci on a chromosome in different members of the population. A chromosome containing the two advantageous alleles can be produced within a few generations by recombination. However, should the same two alleles arise in different members of an asexual population, the only way that one chromosome can develop the other allele is to independently gain the same mutation, which would take much longer. Several studies have addressed counterarguments, and the question of whether this model is sufficiently robust to explain the predominance of sexual versus asexual reproduction remains. 1773-86, 16-18. Ronald suggested that sex might facilitate the spread of advantageous genes by allowing them to better escape their genetic surroundings, if they should arise on a chromosome with deleterious genes. Sources of these theories respond to the balance argument that the individuals produced by sexual and asexual reproduction may differ in other respects too, which may influence the persistence of sexuality. For example, in the heterogamous water fleas of the genus Cladocera, sexual offspring form eggs which are better able to survive the winter versus those the fleas produce asexually. Increased resistance to parasites. One of the most widely discussed theories to explain the persistence of sex is that it is maintained to assist sexual individuals in resisting parasites, also known as the Red Queen hypothesis. 1917, 113-117, 2021-22. When an environment changes, previously neutral or deleterious alleles can become favorable. If the environment changed sufficiently rapidly, i.e. between generations, these changes in the environment can make sex advantageous for the individual. Such rapid changes in environment are caused by the co-evolution between hosts and parasites. Imagine, for example, it's one parasites with two alleles P and P conferring two types of parasitic ability, and one gene in hosts with two alleles H and H, conferring two types of parasite resistance, such that parasites with allele P can attach themselves to hosts with the allele H, and P to H. Such a situation will lead to cyclic changes in allele frequency. As P increases in frequency, H will be disfavored. In reality, there will be several genes involved in the relationship between hosts and parasites. In a sexual population of hosts, offspring will only have a different parasitic resistance if a mutation arises. In a sexual population of hosts, however, offspring will have a new combination of parasitic resistance alleles. In other words, like Lewis Carroll's Red Queen, sexual hosts are continually running, adapting to stay in one place, resist parasites. Evidence for the evolution of sex is provided by comparison of the rate of molecular evolution of genes for kinases and immunoglobulins in the immune system with genes coding other proteins. The genes coding for immune system proteins evolve considerably faster. 23-24. Further evidence Red Queen hypothesis was provided by observing long-term dynamics and parasite coevolution in a mixed, sexual and asexual population of snails Potamopyrgus antipodarum. The number of sexuals, the number of asexuals, and the rates of parasite infection for both were monitored. It was found that clones that were plentiful at the beginning of the study became more susceptible to parasites over time. As parasite infections increased, the once plentiful clones dwindled dramatically in number. Some clonal types disappeared entirely. Meanwhile, sexual snail populations remained much more stable over time. 25-26. However, Hanley et al. 27 studied mite infestations of a parthenogenetic gecko species and its two related sexual ancestral species. Contra expectation based on the Red Queen hypothesis, they found that the prevalence, abundance and mean intensity of mites in sexual geckos was significantly higher than in asexuals sharing the same habitat. In 2011, used the microscopic roundworm Caenorhabditis elegans as a host in the pathogenic bacteria Serratia marcescens to generate a host-parasite coevolutionary system in a controlled environment, allowing them to conduct more than 70 evolution experiments testing the Red Queen hypothesis. They genetically manipulated the mating system of C. elegans, causing populations to mate either sexually, by self-fertilization, or a mixture of both within the same population. Then they exposed those populations to the S. marcescens parasite. It was found that the self-fertilizing populations of C. elegans were rapidly driven extinct by the coevolving parasites while sex allowed populations to keep pace with their parasites, a result consistent with the Red Queen hypothesis. 29. In natural populations of C. elegans, self-fertilization is the predominant mode of reproduction, but infrequent outcrossing events occur at a rate of about 1%. 30. Critics of the Red Queen hypothesis question whether the constantly changing environment of hosts and parasites is sufficiently common to explain the evolution of sex. In particular, Otto and Neusmer. 31 presented results showing that species interactions, e.g. host versus parasite interactions, typically select against sex. They concluded that, although the Red Queen hypothesis favors sex under certain circumstances, it alone does not account for the ubiquity of sex. Gerstein, 32, further stated that, it seems doubtful to us that strong selection per gene is sufficiently commonplace for the Red Queen hypothesis to explain the ubiquity of sex. 33 reviewed numerous genetic studies on plant disease resistance and failed to uncover a single example consistent with the assumptions of the Red Queen hypothesis. Disadvantages of sex and sexual reproduction. The paradox of existence of sexual reproduction is that though it is ubiquitous in multicellular organisms, there are ostensibly many inherent disadvantages to reproducing sexually when weighed against the relative advantages of alternative forms of reproduction, such as asexual reproduction. Thus, because sexual reproduction abounds in complex multicellular life, there must be some significant benefits to sex and sexual reproduction that compensates for these fundamental disadvantages. Population expansion cost of sex. Among the most limiting disadvantages to the evolution of sexual reproduction by natural selection is that an asexual population can grow much more rapidly than a sexual one with each generation. For example, assume that the entire population of a theoretical species has 100 total organisms consisting of two sexes, i.e. males and females, with 50 male-to-female representation, and that only the females of this species can bear offspring. If all capable members of this population procreated once, a total of 50 offspring would be produced the F1 generation. Contrast this outcome with an asexual species, in which each and every member of an equally sized 100 organism population is capable of bearing young. If all the members of this asexual population procreated once, a total of 100 offspring would be produced, twice as many as produced by the sexual population in a single generation. This diagram illustrates the twofold cost of sex. Each individual to contribute to the same number of offspring. 2. The sexual population remains the same size each generation, where the asexual population doubles in size each generation. This idea is sometimes referred to as the twofold cost of sexual reproduction. It was first described mathematically by John Maynard Smith. 30 For his manuscript, Smith further speculated on the impact of an asexual mutant arising in a sexual population, which suppresses meiosis and allows eggs to develop into offspring genetically identical to the mother by meiotic division. 3. The mutant asexual lineage would double its representation in the population each generation, all else being equal. Technically, the problem above is not one of sexual reproduction but of having a subset of organisms incapable of bearing offspring. Indeed, some multicellular organisms, isogamous, engage in sexual reproduction but all members of the species are capable of bearing offspring. 36 The twofold reproductive disadvantage assumes that males contribute only genes to their offspring and sexual females spend half their reproductive potential on sons. Thus, in this formulation, the principal cost of sex is that males and females must successfully copulate, which almost always involves expending energy to come together through time and space. Asexual organisms need not expend the energy necessary to find a mate. Selfish cytoplasmic genes. Sexual reduction implies that chromosomes and alleles segregate and recombine in every generation, but not all genes are transmitted together to the offspring. 35 There is a chance of spreading mutants that cause unfair transmission at the expense of their non-mutant colleagues. These are referred to as selfish because they promote their own spread at the cost of alternative alleles or of the host organism. They include nuclear meiotic drivers and selfish cytoplasmic genes. Meiotic drivers are genes that distort meiosis to produce gametes containing themselves more than the 50% of the time expected by chance. A cytoplasmic gene is a gene located in an organelle, plasmid or intracellular parasite that modifies reproduction to cause its own increase at the expense of the cell or organism that carries it. 35. Genetic heritability cost of sex. A sexually reproducing organism only passes on approximately 50% of its own genetic material to each L2 offspring. This is a consequence of the fact that gametes from sexually reproducing species are haploid. Again, however, this is not applicable to all sexual organisms. There are numerous species which are sexual but do not have a genetic loss problem because they do not produce males or females. Yeast, for example, are isogamous sexual organisms which have two mating types which fuse and recombine their haploid genomes. Both sexes reproduce during the haploid and diploid stages of their life cycle and have a 100% chance of passing their genes into their offspring. 36. Some species avoid the 50% cost of sexual reproduction, although they have sex, in the sense of genetic recombination. In these species, e.g., bacteria, ciliates, dinoflagellates and diatoms, sex and reproduction occur separately. 37-38. DNA repair and complementation. As discussed in the earlier part of this article, sexual reproduction is conventionally explained as an adaptation for producing genetic variation through allelic recombination. As acknowledged above, however, serious problems with this explanation have led many biologists to conclude that the benefit of sex is a major unsolved problem in evolutionary biology. An alternative, informational, approach to this problem has led to the view that the two fundamental aspects of sex, genetic recombination and outcrossing, are adaptive responses to the two major sources of noise in transmitting genetic information. Genetic noise can occur as either physical damage to the genome, e.g. chemically altered bases of DNA or breaks in the chromosome, or replication errors, mutations. 9-13-14. This alternative view is referred to as the repair and complementation hypothesis, to distinguish it from the traditional variation hypothesis. The repair and complementation hypothesis assumes that genetic recombination is fundamentally a DNA repair process, and that when it occurs during meiosis it is an adaptation for repairing the genomic DNA which is passed on to progeny. Recombinational repair is the only repair process known which can accurately remove double-strand damages in DNA, and such damages are both common in nature and ordinarily lethal if not repaired. For instance, double-strand breaks in DNA occur about 50 times per cell cycle in human cells, see naturally occurring DNA damage. Recombinational repair is prevalent from the simplest viruses to the most complex multicellular eukaryotes. It is effective against many different types of genomic damage, and in particular is highly efficient at overcoming double-strand damages. Studies of the mechanism of meiotic recombination indicate that meiosis is an adaptation for repairing DNA. 40. These considerations form the basis for the first part of the repair and complementation hypothesis. In some lines from the earliest organisms, the diploid stage of the sexual cycle, which was at first transient, became the predominant stage, because it allowed complementation, the masking of deleterious recessive mutations, i.e. hybrid vigor or heterosis. Outcross, the second fundamental aspect of sex, is maintained by the advantage of masking mutations and the disadvantage of inbreeding, mating with a close relative, which allows expression of recessive mutations commonly observed as inbreeding depression. Charles, 41, who concluded that the adaptive advantage of sex is hybrid vigor, or as he put it, the offspring of two individuals, especially if their progenitors have been subjected to very different conditions, have a great advantage in height, weight, constitutional vigor and fertility over the self-fertilized offspring from either one of the same parents. However, outcrossing may be abandoned in favor of parthenogenesis or selfing, which retain the advantage of meiotic recombinational repair, under conditions in which the costs of mating are very high. For instance, costs of mating are high when individuals are rare in a geographic area, such as when there has been a forest fire and the individuals entering the burned area are the initial ones to arrive. At such times mates are hard to find, and this favors parthenogenic species. In the repair and complementation hypothesis, the removal of DNA damage by recombinational repair produces a new, less deleterious form of informational noise, allelic recombination, as a byproduct. This informational noise generates genetic variation, viewed by some as the major effect of sex, as discussed in the earlier parts of this article. Deleterious mutation clearance. Mutations can have many different effects upon an organism. It is generally believed that the majority of non-neutral mutations are deleterious, which means that they will cause a decrease in the organism's overall fitness. 42. If a mutation has a deleterious effect, it will then usually be removed from the population by the process of natural selection. Sexual reproduction is believed to be more efficient than asexual reproduction in removing those mutations from the genome. 43. There are two main hypotheses which explain how sex may act to remove deleterious genes from the genome. Evading harmful mutation buildup. While DNA is able to recombine to modify alleles, DNA is also susceptible to mutations within the sequence that can affect an organism in a negative manner. Asexual organisms do not have the ability to recombine their genetic information to form new and differing alleles. Once it occurs in the DNA or other genetic carrying sequence, there is no way for the mutation to be removed from the population until another mutation occurs that ultimately deletes the primary mutation. This is rare among organisms. Hermann Joseph Muller introduced the idea that mutations build up in asexual reproducing organisms. Muller described this occurrence by comparing the mutations that accumulate as a ratchet. Each mutation that arises in asexually reproducing organisms turns the ratchet once. The ratchet is unable to be rotated backwards, only forwards. The next mutation that occurs turns the ratchet once more. Additional mutations in a population continually turn the ratchet, and the mutations, mostly deleterious, continually accumulate without recombination. 34. These mutations are passed on to the next generation because the offspring are exact genetic clones of their parents. The genetic load of organisms in their populations will increase due to the addition of multiple deleterious mutations and decrease the overall reproductive success and fitness. For sexually reproducing populations, studies have shown that single-celled bottlenecks are beneficial for resisting mutation buildup. Passaging a population through a single-celled bottleneck involves the fertilization event occurring with haploid sets of DNA, forming one fertilized cell. For example, humans undergo a single-celled bottleneck in that the haploid sperm fertilizes the haploid egg, forming the diploid zygote, which is unicellular. For example, humans undergo a single-celled bottleneck in that the haploid sperm fertilizes the haploid egg, forming the diploid zygote, which is unicellular. This passage through a single cell is beneficial in that it lowers the chance of mutations from being passed on through multiple individuals. Instead, the mutation is only passed on to one individual. 45 Further studies using Dictyostelium discoideum suggest that this unicellular initial stage is important for resisting mutations due to the importance of high relatedness. High individuals are more closely related, and more clonal, whereas less related individuals are less so, increasing the likelihood that an individual in a population of low relatedness may have a detrimental mutation. Related populations also tend to thrive better than lowly related because the cost of sacrificing an individual is greatly offset by the benefit gained by its relatives and in turn, its genes, according to kin selection. The studies of discoideum showed that conditions of high relatedness resisted mutant individuals more effectively than those of low relatedness, suggesting the importance of high relatedness to resist mutations from proliferating. 46. Removal of deleterious genes. Diagram illustrating different relationships between numbers of mutations and fitness. Condrashav's model requires synergistic epistasis, which is represented by the red line 47-48. Each subsequent mutation has a disproportionately large effect on the organism's fitness. This hypothesis was proposed by Alexei Kondrashev, and is sometimes known as the deterministic mutation hypothesis. 43. It assumes that the majority of deleterious mutations are only slightly deleterious, and affect the individual such that the introduction of each additional mutation has an increasingly large effect on the fitness of the organism. This relationship between number of mutations and fitness is known as synergistic epistasis. By way of analogy, think of a car with several minor faults. Each is not sufficient alone to prevent the car from running, but in combination, the faults combine to prevent the car from functioning. Similarly, an organism may be able to cope with a few defects, but the presence of many mutations could overwhelm its backup mechanisms. Kondrashev argues that the slightly deleterious nature of mutations means that the population will tend to be composed of individuals with a small number of mutations. Sex will act to recombine these genotypes, creating some individuals with fewer deleterious mutations, and some with more. Because there is a major selective disadvantage to individuals with more mutations, these individuals die out. In essence, sex compartmentalizes the deleterious mutations. There has been much criticism of Kondrashev's theory, since it relies on two key restrictive conditions. The first requires that the rate of deleterious mutation should exceed 1 per genome per generation in order to provide a substantial advantage for sex. While there is some empirical evidence for it, for example in Drosophila 49 and E. coli 50, there is also strong evidence against it. For instance, for the sexual species Saccharomyces cerevisiae, yeast, and Neurospora crassa fungus, the mutation rate per genome per replication are 0.0027 and 0.0030 respectively. For nematode worm Caenorhabditis elegans, the mutation rate per effective genome per sexual generation is 0.036. Fifth and secondly, there should be strong interactions among loci, synergistic epistasis, a mutation fitness relation for which there is only limited evidence. 52-53 Conversely, there is also the same amount of evidence that mutations show no epistasis, purely additive model, or antagonistic interactions. Each additional mutation has a disproportionately small effect. Other explanations. Geodecayan's evolutionary theory of sex. Geodecayan suggested that sexual dimorphism provides a partitioning of a species' phenotypes into at least two functional partitions, a female partition that secures beneficial features of the species and a male partition that emerged in species with more variable and unpredictable environments. The partition is suggested to be an experimental part of the species that allows the species to expand their ecological niche, and to have alternative configurations. This theory underlines the higher variability and higher mortality in males, in comparison to females. This functional partitioning also explains the higher susceptibility to disease in males, in comparison to females, and therefore includes the idea of protection against parasites, as another functionality of male sex. Geodecayan's evolutionary theory of sex was developed in Russia in 1960-1980 and was not known to the West till the era of the Internet. Trofimova, who analyzed psychological sex differences, hypothesized that the male sex might also provide a redundancy pruning function. 54. Speed of evolution. Ilan Eshel suggested that sex prevents rapid evolution. He suggests that recombination breaks up favorable gene combinations more often than it creates them, and sex is maintained because it ensures selection is longer term than in asexual populations, so the population is less affected by short-term changes. 785-86, 55. This explanation is not widely accepted, as its assumptions are very restrictive. It has recently been shown in experiments with Chlamydomonas algae that sex can remove the speed limit on evolution. 56. An information analysis using a simplified but useful model shows that in asexual reproduction, the information gain per generation of a species is limited to 1 bit per generation, while in sexual reproduction, the information gain is bounded by, whereas the size of the genome in bits. 57. Libertine bubble theory. The evolution of sex can alternatively be described as a kind of gene exchange that is independent from reproduction. 58. According to the Thierry Laudes, libertine bubble theory, sex originated from an archaic gene transfer process among prebiotic bubbles. 59-60 The contact among the prebiotic bubbles could, through simple food or parasitic reactions, promote the transfer of genetic material from one bubble to another. That interactions between organisms be in balance appear to be a sufficient condition to make these interactions evolutionarily efficient, i.e. to select bubbles that tolerate these interactions libertine, bubbles through a blind evolutionary process of self-reinforcing gene correlations and compatibility. 61. The libertine bubble theory proposes that meiotic sex evolved in proto-eukaryotes to solve a problem that bacteria did not have, namely a large amount of DNA material, occurring in an archaic step of protocell formation and genetic exchanges. Rather than providing selective advantages through reproduction, sex could be thought of as a series of separate events which combine step by step some very weak benefits of recombination, meiosis, gametogenesis and syngamy. 64. Current sexual species could be descendants of primitive organisms that practiced more stable exchanges in the long term, while asexual species have emerged, much more recently in evolutionary history, from the conflict of interest resulting from anisogamy. Parasites and Muller's ratchet. R. Stephen Howe and Curtis Lively were the first to suggest that the combined effects of parasitism and mutation accumulation can lead to an increased advantage to sex under conditions not otherwise predicted. Nature, 1994. Using simulations, they showed that when the two mechanisms act simultaneously, the advantage to sex over asexual reproduction is larger than for either factor operating alone. Origin of sexual reproduction. Many protists reproduce sexually, as do many multicellular plants, animals, and fungi. In the periodic fossil record, sexual reproduction first appeared about 2.0 billion years ago in the Proterozoic eon, 63-64, although a later date, 1.2 billion years ago, has also been presented. 65-66 Nonetheless, all sexually reproducing eukaryotic organisms likely derive from a single-celled common ancestor. 67-59 It is probable that the evolution of sex was an integral part of the evolution of the first eukaryotic cell. 60-69 There are a few species which have secondarily lost this feature, such as Beteloidea and some parthenocarpic plants. Diploidy. Organisms need to replicate their genetic material in an efficient and reliable manner. The necessity to repair genetic damage is one of the leading theories explaining the origin of sexual reproduction. Individuals can repair a damaged section of their DNA via homologous recombination, since there are two copies of the gene in the cell, and if one copy is damaged, the other copy is unlikely to be damaged at the same site. A harmful damage in a haploid individual, on the other hand, is more likely to become fixed, i.e. permanent, since any DNA repair mechanism would have no source from which to recover the original undamaged sequence. 9. The most primitive form of sex may have been one organism with damaged DNA replicating an undamaged strand from a similar organism in order to repair itself. 70. Meiosis. Sex friction appears to have arisen very early in eukaryotic evolution, implying that the essential features of meiosis were already present in the last eukaryotic common ancestor. 67-72 In extant organisms, proteins with central functions in meiosis are similar to key proteins in natural transformation in bacteria and DNA transfer in archaea. 72 For example, RECA recombinase, that catalyzes the key functions of DNA homology search and strand exchange in the bacterial sexual process of transformation, has orthologs in eukaryotes that perform similar functions in meiotic recombination. 72. Natural transfer in bacteria, DNA transfer in archaea, and meiosis in eukaryotic microorganisms are induced by stressful circumstances such as overcrowding, resource depletion, and DNA-damaging conditions. 72-73 This suggests that these sexual processes are adaptations for dealing with stress, particularly stress that causes DNA damage. In bacteria, these stress induce an altered physiologic state, termed competence, that allows active take-up of DNA from a donor bacterium and the integration of this DNA into the recipient genome. See natural competence, allowing recombinational repair of the recipient's damaged DNA. 74. Environmental stresses of DNA damage were a persistent challenge to the survival of early microorganisms, then selection would likely have been continuous through the prokaryote to eukaryote transition. 62-72 And adaptative adjustments would have followed a course in which bacterial transformation or archaeal DNA transfer naturally gave rise to sexual reproduction in eukaryotes. Virus-like RNA-based origin. Sex might also have been present even earlier, in the hypothesized RNA world that preceded DNA cellular life forms. 75. One proposed origin of sex in the RNA world was based on the type of sexual interaction that is known to occur in extant single-stranded segmented RNA viruses, such as influenza virus, and in extant double-stranded segmented RNA viruses such as reovirus. 76. Exposure to conditions that cause RNA damage could have led to blockage of replication and death of these early RNA life forms. Sex would have allowed reassortment of segments between two individuals with damaged RNA, permitting undamaged combinations of RNA segments to come together, thus allowing survival. Such a regeneration phenomenon, known as multiplicity reactivation, occurs in the influenza virus 77 and reovirus. 78. Parasitic DNA elements. The theory is that sexual reproduction originated from selfish parasitic genetic elements that exchange genetic material, that is, copies of their own genome, for their transmission and propagation. In some organisms, sexual reproduction has been shown to enhance the spread of parasitic genetic elements, e.g. yeast, filamentous fungi. 79. Bacterial conjugation is a form of genetic exchange that some sources describe as sex, but technically is not a form of reproduction, even though it is a form of horizontal gene transfer. However, it does support the selfish gene part theory, since the gene itself is propagated through the F plasmid. 70. The cellular origin of sexual reproduction is proposed to have evolved in ancient Holoarchaea as a combination of two independent processes, jumping genes and plasmid swapping. 80. Partial predation. A third theory is that sex evolved as a form of cannibalism. One primitive organism ate another one, but instead of completely digesting it, some of the eaten organism's DNA was incorporated into the DNA of the eater. 70-68. Vaccination-like process. Sex may also be derived from another prokaryotic process. A comprehensive theory, origin of sex as vaccination, proposes that eukaryon sex's syngamy fusion sex arose from prokaryon unilateral sex as infection, when infected hosts began swapping nuclearized genomes containing co-evolved, vertically transmitted symbionts that provided protection against horizontal superinfection by other, more virulent symbionts. Consequently, sex as meiosis fission sex would evolve as a host strategy for uncoupling from, and thereby render impotent, the acquired symbiotic parasitic genes. 81. Mechanistic origin of sexual reproduction. While theories positing fitness benefits that led to the origin of sex are often problematic, several theories addressing the emergence of the mechanisms of sexual reproduction have been proposed. Viral eukaryogenesis. Viral eukaryogenesis theory proposes that eukaryotic cells arose from a combination of a lysogenic virus, an archaean, and a bacterium. The model suggests that the nucleus originated when the lysogenic virus incorporated genetic material from the archaean and the bacterium and took over the role of information storage for the amalgam. The archaea transferred much of its functional genome to the virus during the evolution of cytoplasm, but retained the function of gene translation and general metabolism. The bacterium transferred most of its functional genome to the virus as it transitioned into a mitochondrion. 82. These transformations to lead to the eukaryotic cell cycle. The Vey hypothesis specifies a pox-like virus as the lysogenic virus. A pox-like virus is a likely ancestor because of its fundamental similarities with eukaryotic nuclei. These include a double-stranded DNA genome, a linear chromosome with short telomeric repeats, a complex membrane-bound capsid, the ability to produce capped mRNA, and the ability to export the capped mRNA across the viral membrane into the cytoplasm. The presence of a lysogenic pox-like virus ancestor explains the development of meiotic division, an essential component of sexual reproduction. 83. Meiotic division in the Vey hypothesis arose because of the evolutionary pressures placed on the lysogenic virus as a result of its inability to enter into the lytic cycle. This selective pressure resulted in the development of processes allowing the viruses to spread horizontally throughout the population. The outcome of this selection was cell-to-cell fusion. This is distinct from the conjugation methods used by bacterial plasmids under evolutionary pressure, with important consequences. 82. The possibility of this kind of fusion is supported by the presence of fusion proteins in the envelopes of the pox viruses that allow them to fuse with host membranes. These proteins could have been transferred to the cell membrane during viral reproduction, enabling cell-to-cell fusion between the virus host and an uninfected cell. The theory proposes meiosis originated from the fusion between two cells infected with related but different viruses which recognized each other as uninfected. After the fusion of the two cells, incompatibilities between the two viruses result in a meiotic-like cell division. 83. The two viruses established in the cell would initiate replication in response to signals from the host cell. A mitosis-like cell cycle would proceed until the viral membranes dissolved, at which point linear chromosomes would be bound together with centromeres. The homologous nature of the two viral centromeres would incite the grouping of both sets into tetrads. It is speculated that this grouping may be the origin of crossing over, characteristic of the first division in modern meiosis. The partitioning apparatus of the meiotic-like cell cycle the cells used to replicate independently would then pull each set of chromosomes to one side of the cell, still bound by centromeres. These centromeres prevent their replication in subsequent division, resulting in 4 daughter cells with one copy of one of the two original pox-like viruses. The process resulting from combination of two similar pox viruses within the same host closely mimics meiosis. 83. Neomarin revolution. An alternative theory, proposed by Thomas Cavalier-Smith, was labeled the Neomarin revolution. The designation, Neomarin revolution, refers to the appearances of the common ancestors of eukaryotes and archaea. Cavalier-Smith proposes that the first neomarins emerged 850 million years ago. Molecular biologists assume that this group appeared much earlier, but Cavalier-Smith dismisses these claims because they are based on the, theoretically and empirically, unsound model of molecular clocks. Cavalier-Smith's theory of the Neomarin revolution has implications for the evolutionary history of the cellular machinery for recombination and sex. It suggests that machinery evolved in two distinct bouts separated by a long period of stasis. First, the appearance of recombination machinery in a bacterial ancestor which was maintained for 3 billion years until the Neomarin revolution when the mechanics were adapted to the presence of nucleosomes. The archaeal products of the revolution maintained recombination machinery that was essentially bacterial, whereas the eukaryotic products broke with this bacterial continuity. They introduced cell fusion and ploidy cycles into cell life histories. Cavalier-Smith argues that both bouts of mechanical evolution were motivated by similar selective forces, the need for accurate DNA replication without loss of viability. 84. Questions. Some questions biologists have attempted to answer include: Asexual reproduction exists, if in many organisms it has a 50% cost fitness disadvantage in relation to asexual reproduction. 37. Did mating types of gametes, according to their compatibility, arise as a result of anisogamy, gamete dimorphism, or did mating types evolve before anisogamy? 85-86. Why do most sexual organisms use a binary mating system? Grouping itself offers a survival advantage. A binary recognition-based system is the most simple and effective method in maintaining species grouping. 87. Why do some organisms have gamete dimorphism? Sexual reproduction. Sexual reproduction is a reproduction that involves a complex life cycle in which a gamete, haploid reproductive cells, such as a sperm or egg cell with a single set of chromosomes combines with another gamete to produce a zygote that develops into an organism composed of cells with two sets of chromosomes diploid. One is typical in animals, though the number of chromosome sets and how that number changes in sexual reproduction varies, especially among plants, fungi, and other eukaryotes. 2, 3. In the stage of sexual reproduction, meiosis, the number of chromosomes is reduced from a diploid number, 2n, to a haploid number, n. During fertilization, haploid gametes come together to form a diploid zygote, and the original number of chromosomes is restored. In placental mammals, sperm cells exit the penis through the male urethra and enter the vagina during copulation, 4, 5, while egg cells enter the uterus through the oviduct. Other vertebrates of both sexes possess a cloaca for the release of sperm or egg cells. Sexual reproduction is the most common life cycle in multicellular eukaryotes, such as animals, fungi and plants. 6, 7. Sexual reproduction also occurs in some unicellular eukaryotes. 2, 8. Sexual reproduction does not occur in prokaryotes, unicellular organisms without cell nuclei, such as bacteria and archaea. Processes in bacteria, including bacterial conjugation, transformation and transduction, may be considered analogous to sexual reproduction in that they incorporate new genetic information. 9. Proteins and other features that are key for sexual reproduction may have arisen in bacteria, but sexual reproduction is believed to have developed in an ancient eukaryotic ancestor. 10. In eukaryotes, diploid precursor cells divide to produce haploid cells in a process called meiosis. In meiosis, DNA is replicated to produce a total of 4 copies of each chromosome. This is followed by 2 cell divisions to generate haploid gametes. After DNA is replicated in meiosis, the homologous chromosomes pair up so that their DNA sequences are aligned with each other. During this period before cell divisions, genetic information is exchanged between homologous chromosomes in genetic recombination. Homologous chromosomes contain highly similar but not identical information, and by exchanging similar but not identical regions, genetic recombination increases genetic diversity among future generations. 11. Sexual reproduction. 2 haploid gametes combine into one diploid cell known as a zygote in a process called fertilization. The nuclei from the gametes fuse, and each gamete contributes half of the genetic material of the zygote. Multiple cell divisions by mitosis without change in the number of chromosomes then develop into a multicellular diploid phase or generation. In plants, the diploid phase, known as the sporophyte, produces spores by meiosis. These spores then germinate and divide by mitosis to form a haploid multicellular phase, the gametophyte, which produces gametes directly by mitosis. This life cycle, involving alternation between 2 multicellular phases, the sexual haploid gametophyte and asexual diploid sporophyte, is known as alternation of generations. Sexual reproduction is considered paradoxical. 12. Because asexual reproduction should be able to outperform it as every young organism created can bear its own young. This implies that an asexual population has an intrinsic capacity to grow more rapidly with each generation. 13. This 50% cost is a fitness disadvantage of sexual reproduction. 14. The twofold cost of sex includes this cost and the fact that any organism can only pass on 50% of its own genes to its offspring. However, one definite advantage of sexual reproduction is that it increases genetic diversity and impedes the accumulation of harmful genetic mutations. 15. 11. Selection is a mode of natural selection in which some individuals out-reproduce others of a population because they are better at securing mates for sexual reproduction. 16. 17. It has been described as a powerful evolutionary force that does not exist in asexual populations. 18. Evolution. The first fossilized evidence of sexual reproduction in eukaryotes is from the Stenian period, about 1.05 billion years old. 19. 20. Biologists studying evolution propose several explanations for the development of sexual reproduction and its maintenance. These reasons include the likelihood of the accumulation of deleterious mutations, increasing rate of adaptation to changing environments, 21 dealing with competition, DNA repair, masking deleterious mutations, and reducing genetic variation on the genomic level. 22, 23, 24, 25. All of these ideas about why sexual reproduction has been maintained are generally supported, but ultimately the size of the population determines if sexual reproduction is entirely beneficial. Larger populations appear to respond more quickly to some of the benefits obtained through sexual reproduction than do smaller population sizes. 26. However, newer models presented in recent years suggest a basic advantage for sexual reproduction in slowly reproducing complex organisms. Sexual reproduction allows these species to exhibit characteristics that depend on the specific environment that they inhabit, and the particular survival strategies that they employ. 27. Sexual selection. In order to reproduce sexually, both males and females need to find a mate. Generally in animals, mate choice is made by females while males compete to be chosen. This leads organisms to extreme efforts in order to reproduce, such as combat and display, or produce extreme features caused by a positive feedback known as a Fisherian runaway. Thus sexual reproduction, as a form of natural selection, has an effect on evolution. Sexual dimorphism is where the basic phenotypic traits vary between males and females of the same species. Found in both sex organs and in secondary sex characteristics, body size, physical strength and morphology, biological ornamentation, behavior and other bodily traits. However, sexual selection is only implied over an extended period of time leading to sexual dimorphism. 28. Animals. Arthropods. Aphid giving birth to live young from an unfertilized egg. Harvestman mating. Few arthropods, such as barnacles, are hermaphroditic, that is, each can have the organs of both sexes. However, individuals of most species remain of one sex their entire lives. 29. Species of insects and crustaceans can reproduce by parthenogenesis, especially if conditions favor a population explosion. However, most arthropods rely on sexual reproduction, and parthenogenetic species often revert to sexual reproduction when conditions become less favorable. 30. The ability to undergo meiosis is widespread among arthropods, including both those that reproduce sexually and those that reproduce parthenogenetically. 31. Although this is a major characteristic of arthropods, understanding of its fundamental adaptive benefit has long been regarded as an unresolved problem. 32. That appears to have remained unsettled. Aquatic arthropods may breed by external fertilization, as for example horseshoe crabs do. 33. Or by internal fertilization, where the ova remain in the female's body and the sperm must somehow be inserted. All known terrestrial arthropods use internal fertilization. Husband millipedes, and some crustaceans use modified appendages such as gonopods or penises to transfer the sperm directly to the female. However, most male terrestrial arthropods produce spermatophores, waterproof packets of sperm, which the females take into their bodies. A few such species rely on females to find spermatophores that have already been deposited on the ground, but in most cases males only deposit spermatophores when complex courtship rituals look likely to be successful. A few such species rely on females to find spermatophores that have already been deposited on the ground, but in most cases males only deposit spermatophores when complex courtship rituals look likely to be successful. 29. The nauplius larva of a pennaid shrimp. Arthropods lay eggs. 29. But scorpions are ovoviviparous. They produce live young after the eggs have hatched inside the mother, and are noted for prolonged maternal care. 34. Newly born arthropods have diverse forms, and insects alone cover the range of extremes. Some hatch as apparently miniature adults, direct development, and in some cases, such as silverfish, the hatchlings do not feed and may be helpless until after their first molt. Many hatch as grubs or caterpillars, which do not have segmented limbs or hardened cuticles, and metamorphose into adult forms by entering an inactive phase in which the larval tissues are broken down and reused to build the adult body. 35. Dragonfly larvae have the typical cuticles and jointed limbs of arthropods but are flightless water breathers with extendable jaws. 36. Crustaceans commonly hatch as tiny nauplius larvae that have only 3 segments and pairs of appendages. 29. Insects. Australian emperor dragonfly laying eggs, guarded by a male. Insect species make up more than 2/3 of all extant animal species. Most insect species reproduce sexually, though some species are facultatively parthenogenetic. Many insect species have sexual dimorphism, while in others the sexes look nearly identical. Typically they have 2 sexes with males producing spermatozoa and females ova. The ova develop into eggs that have a covering called the chorion, which forms before internal fertilization. In diverse mating and reproductive strategies, most often resulting in the male depositing a spermatophore within the female, which she stores until she is ready for egg fertilization. Fertilization, and the formation of a zygote, and varying degrees of development. In many species the eggs are deposited outside the female, while in others, they develop further within the female and the young are born live. 37. Mammals. Genitourinary system of a male and female cat. There are 3 extant kinds of mammals: monotremes, placentals and marsupials, all with internal fertilization. In placental mammals, offspring are born as juveniles, complete animals with the sex organs present although not reproductively functional. After months or years, depending on the species, the sex organs develop further to maturity and the animal becomes sexually mature. Most female mammals are only fertile during certain periods during their estrus cycle, at which point they are ready to mate. 38. For most mammals, males and females exchange sexual partners throughout their adult lives. 39, 40, 41. Fish. The vast majority of fish species lay eggs that are then fertilized by the male. 40. Some species lay their eggs on a substrate like a rock or on plants, while others scatter their eggs and the eggs are fertilized as they drift or sink in the water column. Some fish species use internal fertilization and then disperse the developing eggs or give birth to live offspring. Fish that have live-bearing offspring include the guppy and mollies or Piscilia. Fish that give birth to live young can be ovoviviparous, where the eggs are fertilized within the female and the eggs simply hatch within the female body, or in seahorses, the male carries the developing young within a pouch, and gives birth to live young. 43. Fishes can also be viviparous, where the female supplies nourishment to the internally growing offspring. Some fish are hermaphrodites, where a single fish is both male and female and can produce eggs and sperm. In hermaphroditic fish, some are male and female at the same time, while in other fish they are serially hermaphroditic, starting as one sex and changing to the other. In at least one hermaphroditic species, self-fertilization occurs when the eggs and sperm are released together. Internal self-fertilization may occur in some other species. 4. One species does not reproduce by sexual reproduction but uses sex to produce offspring. Poecilia formosa is a unisex species that uses a form of parthenogenesis called gynogenesis, where unfertilized eggs develop into embryos that produce female offspring. Poecilia formosa mate with males of other fish species that use internal fertilization. The sperm does not fertilize the eggs but stimulates the growth of the eggs, which develops into embryos. 45. Plants. Plants have life cycles with a single diploid multicellular phase that produces haploid gametes directly by meiosis. Male gametes are called sperm, and female gametes are called eggs or ova. In an animal, fertilization of the ovum by a sperm results in the formation of a diploid zygote that develops by repeated meiotic divisions into a diploid adult. Plants have two multicellular life cycle phases, resulting in an alternation of generations. Plant zygotes germinate and divide repeatedly by mitosis to produce a diploid multicellular organism known as the sporophyte. The sporophyte produces haploid spores by meiosis that germinate and divide by mitosis to form a multicellular gametophyte phase that produces gametes at maturity. The gametophytes of different groups of plants vary in size. Mosses and other pteridophytic plants may have gametophytes consisting of several million cells, while angiosperms have as few as 3 cells in each pollen grain. Flowering plants. Flowers contain the sexual organs of flowering plants. Flowering plants are the dominant plant form on land. 46, 168, 173, and they reproduce either sexually or asexually. Often their most distinctive feature is their reproductive organs, commonly called flowers. The anther produces pollen grains which contain the male gametophytes that produce sperm nuclei. For pollination to occur, pollen grains must attach to the stigma of the female reproductive structure, carpel, where the female gametophytes are located within ovules enclosed within the ovary. After the tube grows through the carpel style, the sex cell nuclei from the pollen grain migrate into the ovule to fertilize the egg cell and endosperm nuclei within the female gametophyte in a process termed double fertilization. The zygote develops into an embryo, while the triploid endosperm, one sperm cell plus 2 female cells, and female tissues of the ovule give rise to the surrounding tissues in the developing seed. The ovary, which produced the female gametophytes, then grows into a fruit, which surrounds the seeds. Plants may either self-pollinate or cross-pollinate. In 2013, flowers dating from the Cretaceous, 100 million years before present, were found encased in amber, the oldest evidence of sexual reproduction in a flowering plant. Microscopic images showed tubes growing out of pollen and penetrating the flower's stigma. The pollen was sticky, suggesting it was carried by insects. 47. Ferns. Ferns produce large diploid sporophytes with rhizomes, roots and leaves. Fertile leaves produce sporangia that contain haploid spores. The spores are released and germinate to produce small, thin gametophytes that are typically heart-shaped and green in color. The gametophyte prothely produce motile sperm in the antheridia and egg cells in archegonia on the same or different plants. 48. After when dew deposits a film of water, the motile sperm are splashed away from the antheridia, which are normally produced on the top side of the thallus, and swim in the film of water to the archegonia where they fertilize the egg. To promote outcropping or cross-fertilization, the sperm are released before the eggs are receptive of the sperm, making it more likely that the sperm will fertilize the eggs of different thallus. After fertilization, a zygote is formed which grows into a new sporophytic plant. The condition of having separate sporophyte and gametophyte plants is called alternation of generations. Bryophytes. The bryophytes, which include liverworts, hornworts and mosses, reproduce both sexually and vegetatively. They are found growing in moist locations and like ferns, have motile sperm with flagella and need water to facilitate sexual reproduction. Plants start as a haploid spore that grows into the dominant gametophyte form, which is a multicellular haploid body with leaf-like structures that photosynthesize. Haploid gametes are produced in antheridia, male, and archegonia, female, by mitosis. The cells released from the antheridia respond to chemicals released by ripe archegonia and swim to them in a film of water and fertilize the egg cells, thus producing a zygote. The zygote divides by meiotic division and grows into a multicellular, diploid sporophyte. The sporophyte produces spore capsules, sporangia, which are connected by stalks, setae, to the archegonia. The spore capsules produce spores by meiosis, and when ripe the capsules burst open to release the spores. Bryophytes show considerable variation in their reproductive structures, and the above is a basic outline. Also in some species each plant is one sex, dioicous, while other species produce both sexes on the same plant, monoicous. 49. Fungi. Puffballs emitting spores. Fungi are classified by the methods of sexual reproduction they employ. The outcome of sexual reproduction most often is the production of resting spores that are used to survive inclement times and to spread. There are typically 3 phases in the sexual reproduction of fungi: plasmagamy, karyogamy and meiosis. The cytoplasm of 2 parent cells fuse during plasmagamy and the nuclei fuse during karyogamy. New haploid gametes are formed during meiosis and develop into spores. The adaptive basis for the maintenance of sexual reproduction in the Ascomycota and Basidiomycota dicarion fungi was reviewed by Wallen and Perlin. They concluded that the most plausible reason for maintaining this capability is the benefit of repairing DNA damage, caused by a variety of stresses, through recombination that occurs during meiosis. 50. Bacteria and archaea. 3 distinct processes in prokaryotes are regarded as similar to eukaryotic sex Bacterial transformation, which involves the incorporation of foreign DNA into the bacterial chromosome. Bacterial conjugation, which is a transfer of plasmid DNA between bacteria, but the plasmids are rarely incorporated into the bacterial chromosome, and gene transfer and genetic exchange in archaea. Bacterial transformation involves the recombination of genetic material, and its function is mainly associated with DNA repair. Bacterial transformation is a complex process encoded by numerous bacterial genes, and is a bacterial adaptation for DNA transfer. 22-23. This process occurs naturally in at least 40 bacterial species. 51. For a bacterium to bind, take up, and recombine exogenous DNA into its chromosome, it must enter a special physiological state referred to as competence. See natural competence. Sexual reproduction in early single-celled eukaryotes may have evolved from bacterial transformation. 24. Or from a similar process in archaea. See below. On the other hand, bacterial conjugation is a type of direct transfer of DNA between two bacteria mediated by an external appendage called the conjugation pilus. Bacterial conjugation is controlled by plasmid genes that are adapted for spreading copies of the plasmid between bacteria. Frequent integration of a plasmid into a host bacterial chromosome, and the subsequent transfer of a part of the host chromosome to another cell do not appear to be bacterial adaptations. 22-53. Exposure of hyperthermal sulfolobus species to DNA-damaging conditions induces cellular aggregation accompanied by high-frequency genetic marker exchange. 54-55. Ajon et al. 55 hypothesized that this cellular aggregation enhances species-specific DNA repair by homologous recombination. DNA sulfolobus may be an early form of sexual interaction similar to the Morrell studied bacterial transformation systems that also involve species-specific DNA transfer leading to homologous recombinational repair of DNA damage. See also Sexy son hypothesis. The sexy son hypothesis in evolutionary biology and sexual selection, proposed by Patrick J. Weatherhead and Raleigh J. Robertson of Queen's University in Kingston, Ontario in 1979, 1. States that a female's ideal mate choice among potential mates is one whose genes will produce males with the best chance of reproductive success. This implies that other benefits the father can offer the mother or offspring are less relevant than they may appear, including his sexy son hypothesis. The sexy son hypothesis in evolutionary biology and sexual selection, proposed by Patrick J. Weatherhead and Raleigh J. Robertson of Queen's University in Kingston, Ontario in 1979, 1. States that a female's ideal mate choice among potential mates is one whose genes will produce males with the best chance of reproductive success. This implies that other benefits the father can offer the mother or offspring are less relevant than they may appear, including his capacity as a parental caregiver, territory and any nuptial gifts. Sexy son hypothesis. The sexy son hypothesis in evolutionary biology and sexual selection, proposed by Patrick J. Weatherhead and Raleigh J. Robertson of Queen's University in Kingston, Ontario in 1979, 1. States that a female's ideal mate choice among potential mates is one whose genes will produce males with the best chance of reproductive success. This implies that other benefits the father can offer the mother or offspring are less relevant than they may appear, including his capacity as a parental caregiver, territory and any nuptial gifts. Fisher's principle means ratio, except in certain eusocial insects, is always near 1 between males and females, yet what matters most are her sexy sons, future breeding successes, more likely if they have a promiscuous father, in creating large numbers of offspring carrying copies of her genes. 2. This sexual selection hypothesis has been researched in species such as the European pied flycatcher. 1. European pied flycatcher. Ronald Fisher in 1912. Context. Mating preferences are widely recognized as being responsible for the rapid and divergent evolution of male secondary sex characteristics. 3. In 1915, Ronald Fisher wrote, 4. While this taste and preference prevails among the females of the species, the males will grow more and more elaborate and beautiful tail feathers. The question must be answered, why have the females this taste? Of what use is it to the species that they should select this seemingly useless ornament? The first step solution lies in the fact that the success of an animal in the struggle for existence is not measured only by the number of offspring which it produces and rears, but also by the probable success of these offspring. So that in selecting a mate from a number of different competitors, it is important to select that one which is most likely to produce successful children. 4. In 1976, prior to Weatherhead and Robertson's paper, 1. Richard Dawkins had written in his book The Selfish Gene. In a society where males compete with each other to be chosen as he-men by females, one of the best things a mother can do for her genes is to make a son who will turn out in his turn to be an attractive he-man. If ensure that her son is one of the fortunate few males who wins most of the copulations in the society when he grows up, she will have an enormous number of grandchildren. The result of this is that one of the most desirable qualities a male can have in the eyes of a female is, quite simply, sexual attractiveness itself. 5. Ronald's principle, as published in his book The Genetical Theory of Natural Selection, is one of several possible explanations for the highly diverse and often astonishing ornaments of animals. 7. 8. If females choose physically attractive males, they will tend to get physically attractive sons, and, thus more grandchildren, because other choosy females will prefer their attractive, sexy sons. The theory will function regardless of the physical or behavioral trait a female chooses, as long as it is heritable, that is, the trait varies between individuals of the population because it is possessing the trait that makes males attractive, and not the qualities of the trait in itself. Once a preference becomes established, females choosing males with elaborate secondary sexual traits will produce sons that carry alleles for the trait and produce daughters that carry alleles for the preference, generating genetic coupling that will drive self-reinforcing coevolution of both trait and preference, due to the mating advantage of males with the trait, creating a Fisherian runaway sexy sons process. 8 similar models have been proposed for postcopulatory female preferences, such as the time at which females remove the male's sperm ampulla after mating. Sexual selection by direct and/or indirect benefits as well as sexual conflict determine the evolution of animal mating systems. 9. Its original context, the narrow-sense sexy son hypothesis of Weatherhead and Robertson refers to mating systems with care from both parents. In these mating systems, females that mate with a polygynous male normally receive less assistance than females mated with a monogamous male, and thus suffer from direct fitness consequences that have to be, at least, compensated for by the breeding successes of their sexy sons. On the other hand, a broad-sense sexy son hypothesis encompasses both polygyny and promiscuous mating systems, with and without care from both parents. Otto 1911 argues that the costs of any additional choice may be so minor that female choice for honestly signaling males, that is good genes, may evolve even if the indirect benefits on offspring quality are small. A similar argument can be made for the sexy son hypothesis if mates of attractive males do not suffer any direct fitness consequences. 12. Sexual conflict. Guion and Cock of the Rock. Sexual conflict refers to the conflicting goals of breeding males and females. It describes the diverging interests of males and females in optimizing their fitness. From the viewpoint of any partner, the best outcome would be for the partner's mate to care for the young, thus freeing up his or her own resources, e.g., time and energy that he, but typically he, can invest in further sex that may create additional offspring. In polygynous mating systems, sexual conflict means the optimization of male reproductive success by having mated with multiple females, even though the reproductive success of a polygynously mated female is thereby reduced. 12. The case for the Guion and Cock of the Rocks, whose male members spend a majority of their time and energy maintaining their plumage and attempting to seek the most matings. Females, on the other hand, spend their time building and maintaining their nest where they will lay their eggs and raise the young. Good genes theory. Good genes theory proposes that females select males seen to have genetic advantages that increase offspring quality. Stability of offspring provides compensation for any lower reproductive success that results from their being picky. The good gene hypothesis for polyandry proposes that when females encounter better males than their previous mates, they remate in order to fertilize their eggs with the better male's sperm. 7. Individuals who have selected mates with better genetics tend to have offspring that survive longer and are more able to reproduce than those that do not pick mates with genetic quality. This suggests that carefully choosing a mate is beneficial. 13. A study notes that pronghorn females engage in an obvious and energetically expensive mate sampling process to identify vigorous males. Though each female selects independently, the outcome is that a small proportion of the herd's males sire most young. Offspring of attractive males were more likely to survive to weaning into age classes as late as 5 years, apparently due to faster growth rates. Because pronghorn males do not have costly ornaments, the authors conclude that female choice for good genes can exist in the absence of obvious sexual selection cues such as elaborate antlers. The sexy son hypothesis is closely related to the good genes assumption in the Fisherian runaway selection process. Like genes, the sexy son hypothesis assumes the existence of indirect genetic benefits that are able to compensate for any inferior direct reproductive success, i.e., fewer offspring. The main difference between good genes and the sexy son hypothesis is that the latter assumes an indirect effect due to the attractiveness of the sons, whereas good genes focus on the viability of both sons and daughters. However, attractiveness is not narrowly defined, and can refer to every trait that increases a male's probability to become polygynous. Sperm models. The successful sperm is the one that fuses with the egg. Good sperm models predict positive genetic associations between a male's sperm competitiveness and the general viability of his offspring. 7. Whereas sexy sperm models predict that multiple mating females produce more grandchildren. As with precopulatory processes, postcopulatory models predict that the trait in males that determines fertilization success will become genetically coupled with the mechanism by which females choose the sperm of preferred males. Sexual conflict. Guion and Cock of the Rock. Sexual conflict refers to the conflicting goals of breeding males and females. Describes the diverging interests of males and females in optimizing their fitness. From the viewpoint of any one partner, the best outcome would be for the partner's mate to care for the young, thus freeing up his or her own resources, e.g., time and energy that he, but typically he, can invest in further sex that may create additional offspring. In polygynous systems, sexual conflict means the optimization of male reproductive success by having mated with multiple females, even though the reproductive success of a polygynously mated female is thereby reduced. This can be the case for the Guion and Cock of the Rocks, whose male members spend a majority of their time and energy maintaining their plumage and attempting to seek the most matings. Females, on the other hand, spend their time building and maintaining their nest where they will lay their eggs and raise the young. Good genes theory. Genes theory proposes that females select males seen to have genetic advantages that increase offspring quality. Increased viability of offspring provides compensation for any lower reproductive success that results from their being picky. Gene hypothesis for polyandry proposes that when females encounter better males than their previous mates, they remate in order to fertilize their eggs with the better male's sperm. 7. Dungles who have selected mates with better genetics tend to have offspring that survive longer and are more able to reproduce than those that do not pick mates with genetic quality. This suggests that carefully choosing a mate is beneficial. 13. Another study notes that pronghorn females engage in an obvious and